right, hello everybody. Thank you so much for making it tonight. We understand that these are challenging times for many reasons, but we are so excited to have you here tonight and hope that this is a fun educational webinar for all of you. We are here to build RAM community, and we hope that this is just one of the many resources of ways that we can do that. First things first, I would love to thank all of our members for participating tonight. Without you, we would not be able to be hosting events like this. So thank you so much for all of your membership every single year. Thank you for our life members that are here and our 3-year members as well. Events like this are not possible without you. While I am talking and people are slowly logging in right now, just wanted to let you all know that number one, this is a webinar, so we cannot see you or hear you, but we do ask you to use the chat feature use our question and answer feature. We will be answering questions throughout the program, so if you have a question while Dr. Rushika is talking, please feel free to just type it in, and then I'll stop her and we'll ask the question, address the answer, and then move on. Also, while I am talking, if you all would like, you can go to the chat feature and you can change the to aspect from all panelists to all panelists and attendees. And from there, you can type in your name, what year you graduated, where you're from, and what college that you graduated from too. Just a nice way to connect with other RAMs that are here tonight. I would also love to thank all of our sponsors for tonight. Women in Philanthropy have helped find presenters as well as promotions for the event. The Vice President for Research and Development with Colorado State University. We are so excited to be able to partner with them on finding more speakers every single month. And then the College of Veterinary Medicine and Biomechanical Sciences as well. In just a few moments, I'm going to post information about each one of those sponsors and partners of CSU, and that way you guys can read more information about them as well. Without further ado, I would love to stop talking, because I'm sure you guys are all here to meet Dr. Rushika Perera and give her the floor. Just a couple of things about her. She is incredible. She's doing so much work on campus. I believe she's been with CSU for more than 7 years now, which is really awesome to have somebody who is constant and doing this much research on campus. I believe too, I just found this out today, was that she was also named 2020's American Chemical Society Infectious Disease Young Investigator. The longest title ever, so congratulations to you on that. And without further ado, I will let you take it away.
Thank you. I'm going to share my screen. And my computer takes a little time to show me what I'm looking at. Can you all see that?
Yes, you are good to go.
Thank you. So thanks again to the Alumni Association for being here, and also for inviting me to present our work on COVID-19 therapeutics. One of the main concepts that we need to be thinking about is that when viruses infect our human or animal bodies, there's infinite interactions that occur. And so when those infinite interactions occur, our therapeutics or vaccines might be altered in their efficacy if we don't understand what those interactions are. So this work that I'm about to present is not just about therapeutic testing, but it's also about breaking the barriers of how we look at therapeutics. So just to give you some background, SARS-CoV-2 originated in bats. Thousands of coronavirus sequences in many bat species are found every year. There have been 4 significant coronavirus spillover events, and spillover events means that you have viruses from the animal species spilling over to the human species. So if you remember a while ago, many years ago, SARS-CoV-1 came out, but people started showing signs of disease before they shed virus. And so it was easy to get a hold of that epidemic because quarantine brought it to an end. When MERS-CoV spilled over, it mostly spilled over to dromedary camels, and now all outbreaks are originating from these camels. When SARS-CoV spilled over, we were able to control the epidemic by, unfortunately, killing many piglets. And then SARS-CoV-2, unfortunately, transmits prior to disease onset, and so it's really efficient in infection, and so it makes it much more difficult to get a handle of it. It's for sure that we will expect many more spillovers in the future because these bats carry hundreds of coronaviruses. So when SARS-CoV-2 hit and COVID-19 became a pandemic, CSU had to respond to a need. There was a significant need for antiviral testing from all around the world, and CSU set up a 6,400 square foot BSL-3 space, and we converted it into a respiratory virus wing. Sorry, my thing is on a timer, I think. We were able to then start working with people all around the world, testing antivirals and disinfectants and inhalants and biologics, which are like natural products. So our testing has been designed into 3 phases. The first phase is to establish conditions to test drugs and antivirals and therapeutics. Should I get rid of this timer? Sorry. The second phase was to develop synergy models for combination therapeutics. And then phase 3 was to identify new ways of testing therapeutics. So I'll go through all 3 phases. So the first phase is where we used basic virus assays to test drugs. And so we have SARS-CoV-2 isolated from the USA in Washington in January. And we use cells to test these drugs in vitro, so in the lab. These are cells isolated from African green monkey or human cancers, mostly human hepatoma, and some primary cells. Oh, this has a mind of its own. And then we have assays in vitro. So we look at cell death, virus growth, and we also look at visual methods of imaging, as well as genome quantification. So we can also test animal models. We have several colleagues here, Dr. Richard Bowen and Tony Shounds and Rebecca Kading, who have animal models of coronavirus infection. And so we can test these drugs in animal models. So the drugs that we've been using are investigational compounds or FDA approved drugs, natural products, and inhalants, and also eye washers. So, sorry. Corey, can I get out of this and stop the, the timer?
Yeah, you're just gonna have to close down the presentation.
Oh, yeah. Is that okay or no?
Yeah, that's fine.
Okay, otherwise this is gonna go crazy. Sorry about that, everybody. I'll be back.
And again, while she's fixing some of that stuff, I had posted a couple things inside the chat. So feel free to click on those links and hear more about the Alumni Association or other upcoming events. I also have information in there if you get disconnected. Sometimes internet stops or is having an issue. Don't worry, just close down Zoom and restart it as well.
I'm almost there. Okay, can you see me?
Yep, we can see it.
Awesome. Sorry about that. So we've been working with several compounds. One is investigational compounds. So these are drugs that haven't been FDA approved, and they're being explored as potential drugs for the future. There's others that are FDA approved as well. Natural products are basically plants and plant extracts and fruit extracts. So there's many companies around the world, as well as many countries that have a vast array of natural products. And this is becoming a very popular effort to kind of use as antivirals or test as antivirals. Inhalants are basically used in lots of lung diseases like TB, and I'll talk about that a little bit. So we are testing several inhalants because COVID-19 causes lots of lung complications. And then eye washes, where this is mainly for first responders in case they get sprayed in the eye with with SARS coronavirus, they can actually use this eye wash to disinfect their eyes. So what we do is when we have these compounds to test, we add them to cells and we try to pre-treat the cells for about 24 hours. And then we add virus, and then we're able to wait a few days and measure if there's virus growth. So if the drug works, then we should see less virus. And if the drug doesn't work, we see more virus. So the interesting thing about this virus is that it kills cells. And so we can look using a method called the plaque assay to see how many virus particles exist. So what you see here in the dots are basically a count of how many virus particles. So each white spot corresponds to a virus particle. And so we can count how many virus particles are coming out. And if there's a lower number, that means that the drug is working. We can also look at cell death. So what you see here is an image of what live cells look like and what dead cells look like. And this virus kills cells. So your virus infected cells look like this. We can also use an antibody against the viral proteins and be able to look in color as to whether what cells are infected by virus. So what you see here in green is virus infected cells. We also can measure the genome of the virus. So it's kind of like when you get a nasal swab, what they're measuring is basically the genome of the virus. And we can look at how infectious the virus is after drug treatment. So one of the important things for us to start with was, can we do this work in testing these drugs? And can multiple operators produce really good data? So this is an example of two assays. What you see on the y-axis, on the vertical axis, is how much cell death is occurring. And on the x-axis is how much drug. So yeah, as you go to the right, you have increasing amounts of drug. And what you see here is that the cell death, so the cell cytotoxicity decreases with time. And so one of the important things here is to measure that there's two operators, so Elena and myself. So Elena is EL and myself is RP, that we were able to do this experiment and produce the same exact results. And this was important because it shows that we can do the work well. We also wanted to show that we can use drugs that are already tested and we can reproduce the data that has been published. So for instance, you must be familiar with remdesivir. That has been talked about in the news. And we were able to produce, show that remdesivir does actually function to kill the virus. So one of the other aspects that we had to develop was different ways of measuring virus and drug efficacy. So as I said before, we use plaque assays, we use immunofluorescence, and then we can measure the genome. So having multiple methods to measure the same thing really validates your efforts and your data. So this is a busy slide, and I apologize, but it shows something very important. Each of these graphs is a different type of assay to measure the same thing. And what we're measuring here is how well the drug works. So in green is when there's a lot of cells. In green is when there's a lower number of cells, and in orange is when there's a lot of cells. And what we're showing here is the type of assay you use to measure something matters because it's giving you different data. So we were able to optimize that these two assays, the neutral red assay, the AOPI staining assay, actually can give us information about how the drug inhibits the virus better than some of the other assays could. So this is important, and many scientists or many people around the world don't really think about this. And so CSU is really good at its virology and infectious disease. And so we were able to really think about this and develop this. So I'll show you some data on disinfectants and inhalants. So disinfectant is basically what you use to clean surfaces. And as you know, during COVID-19, there's an increased effort to use disinfectants to clean surfaces, your hands, and anything that is that people touch. So we have to test several disinfectants for many companies around the world. And what we do is we mix the disinfectant with virus, and then we look to see how much of virus is killed. And what you can see in this graph is basically saying the green bars are showing that when there's no disinfectant, there's a lot of virus produced. So the height of the bar says that there is a lot of virus produced. But the reagent corresponds to the disinfectant. When the disinfectant is used, we actually kill most of the virus. We were not able to detect any of this virus. So this disinfectant, once we tested it in cells, we were also able to test it in animals, and it worked really well. So it was submitted for FDA approval. So next I will talk about inhalants, and these are inhalants that are used in lung diseases like tuberculosis. And this company was interested in understanding if this inhalant could also kill the virus during COVID-19. So in COVID-19 patients, you see a lot of lung pathology. And the question was, can this inhalant reduce that pathology and kill the virus? So what you see here in red is basically as you increase the concentration of the inhalant, you can decrease the amount of virus. So basically it's showing that the virus is being killed definitely. And it's also showing that there is no pathology due to the inhalant. So your lung tissue is not going to get damaged because of this inhalant. So this inhalant has now gone on to clinical trials in phase 2 in the UK. The nice thing about this company is that they are also willing to work with us to publish these results. Many times companies don't let us publish results because they have proprietary patents, but this company is willing to work with my student to publish the results, so that we really appreciate that as an academic institution. So we've also developed new technologies on how to study antiviral efficacy against viruses. And so next I'm going to show you what that new technology is like. So this is called a Celigo imaging cytometry. And so basically it's a machine that can look at a lot of plates and a lot of wells in a plate all at once. So instead of testing one sample at a time, we can actually test 96 samples at a time. And so what it tells you is you can use different types of antibodies and different types of colors to understand how many cells are dead because of the virus, how many cells are alive, and if the drug is actually increasing that live to dead cell ratio. And this is really helpful when you're testing multiple drugs, because some companies send us hundreds of drugs, and this will expedite our efforts. So this is results from 3 different types of assays. As I said before, we want to test the same thing using multiple methods so that we're really sure about the data that we're providing these companies. So on the left, in blue are dead cells, in green are live cells, and then in brown is the virus. So I just want to point out that as you go to the right, this is increasing amount of drug. And then as you go up, you're increasing the amount of live cells. So what you see here, where I've pointed out in red, is that you have a concentration of the drug where you have reduced cell death and increased live cells. So what that says is that the virus is inhibited, and so it's not killing the cells, so you get more live cells. And in this case, you also see that the virus is down. So this concentration of drug seems to work really well. And on the right-hand side, we validated that by showing that if we measure the amount of virus, that concentration actually reduces the amount of virus that is produced. What has to be said here is that biologics are basically natural products. And so your concentration of the effective chemical that is actually killing the virus is not high. And so that's why you don't see as much of a drop of the virus infection as you would if it's a pure drug. So we can also use antibodies against the virus and look at cells that are infected by the virus. So what you see here on the left are cells from African green monkeys. And these cells are infected by virus. And then we can use the antibody against one of the viral proteins and look using microscopy at what cells are infected and what cells are not infected. And on the three black panels are basically controls saying that our assay actually works. And so this is an important assay because it shows how a virus can spread from one cell to the other, and it gives us more impact on how we can study this. So we're going to continue to test FDA-approved drugs and investigational compounds and lots of other technologically advanced therapeutics that companies are producing. This is done as a service to the community and to the world because we don't, as an academic institution, we're not really allowed to charge very much for the efforts. We're just charging for our materials. So I would like to talk about what we're doing in developing synergy models for combination therapeutics. And what that means is we're trying to really push the envelope of therapeutics and try to understand how combining different therapeutics might be impactful. So the problem with single drugs, as you know, like remdesivir or hydroxychloroquine, is that you need a high dose when you're using a single drug. And that if you're using a high dose, then you have lots of complications, right? So you could be hyper-reactive to the drug. So you could react very strongly to the drug. That would not be very good. You could not react at all to the drug. So you'll be hyper-reactive. And then you could also have idiosyncratic or tachyphylaxis. And what that means is an individual could either not respond to the drug, or the individual could be responding to the drug and suddenly you will not respond to the drug. So there's multiple complications that are involved with single therapies. And if you take a high dose of drug, then maybe you also have comorbidities that are impacted, like heart disease and diabetes. So for the hepatitis C and AIDS diseases, it has been really shown that multiple drugs and therapies actually work. So if you use 2 or 3 drugs at once, you can use less of each, and that has much more powerful impact than if you use a single drug. So we decided to work with this one company to look at combination drugs. So these are 3 FDA-approved drugs. 2 of them you're very familiar with, but I'm unfortunately not allowed to say what they are because of non-disclosure agreements. But you have drug A, drug B, and drug C. And so if you put them in combination, you can have multiple effects, right? One is additive. That means the effect of drug A is added to drug B and drug C, and so you get an additive effect. You might get an antagonistic effect. So drug A could reduce the efficacy of drug B or C and vice versa. So what we really want is synergistic combinations. What that means is if you add drug B to drug A, it will really push the effort of drug A and the efficacy of drug A further than it would normally be alone. So adding drug B and C to drug A really enhances the efficacy of drug A, and that's what we really want to do. So, oh, my animations are not that great. I will show you some data that we have with these triple drugs. So we tested 3 drugs together. And what you see in this graph is on the y-axis, as you go higher, you have a cytopathic effect, which means that the cells are dying. So in blue is just virus only without any drugs. And you can see that it takes anywhere from 0 to 6 days to peak produce virus. Sorry. And then in orange is a single drug. In yellow is drug A plus B. And then in green is drug A plus B plus C. And on the x-axis is days post-infection. So what you see here is that with 3 drugs, you have a 2.5-day delay with the triple drug rather than with the single or individual drugs. And the amount of virus can be measured with the triple drug as well. And we see like 6 logs of reduction in the triple drug concentrations. So one more interesting thing is that we also see that the virus peaks early, and then you have pathology showing up later. And this is what we see in human patients. This is what we see in animal studies as well. So what you see here is in the x-axis are days post-infection, and on the y-axis is cytopathic effect. So with not just virus alone, you peak cytopathic effect, which means that a maximum amount of virus is produced in 2 days. With one drug, that amount of virus produced is reduced. With the double drug, it's reduced even more. But with the triple drug, which is shown in green, you see that there is a significant delay in the amount of virus produced, but also there's a reduction in the amount of virus produced, but there's also a delay in the production of virus. So it's really effective. And what you see on the solid lines is basically how fast are the cells dying. And so again, in green is the triple drug, and you can see that the cell death has been reduced. So what we're showing here is that with a triple drug combination, that we are really delaying the pathology caused by the virus, and we're also reducing the virus. So this has now been sent in for publication, and you can actually, the preprint, the pre-publication is at this website under the BioRx server. And then these triple drugs have also been, are now being tested in clinical trials. So finally, I want to talk about how CSU is really pushing the envelope and paving new roads to identifying better therapeutics. So as you know, this is the human organism, and we are impacted physiologically by many factors, right, in our daily lives. So we can be bitten by mosquitoes, we can have bacterial and viral infections, we get vaccinations and therapeutics, we're affected by the environment, and then also the microbiome, so your probiotics that you take. And so historically, people have been studying how your physiology changes by looking at the genes using genomics, or the RNA using transcriptomics, or proteins using proteomics. And what they're looking here for is, upon having a bacterial infection, what genes in your body are changing, right? And do those genes alter the disease severity due to the bacterial infection? But what is really important are the metabolites that are produced. So these are the results of biochemical pathways and by the results of activation of all these proteins. And they are the ones that cause the phenotype. So if you have a high glucose content in your body, and because of and you're diabetic, that is caused by glucose, which is a metabolite, right? So looking at the metabolites really alters the way that we look at disease in our bodies. And the nice thing is we can look at a snapshot of the human bodies and understand what metabolites exist. So how much sugar do you have today in your blood? And then you can go back and figure out what biochemical pathways are producing that sugar, and then try to shut those biochemical pathways down. So what is important to understand is that metabolites are really powerful molecules that we should measure, because the intermediates or products of active biochemical pathways. They are the closest relationship to a phenotype. So they are the molecules that actually cause the different disease pathology that you see, whether it's fever or other types of damage, tissue damage. They also can be used as biomarkers. So for instance, we can look at your blood and we can measure cholesterol, or we can measure sugar and tell you that your cholesterol is high, and so there's potential for you to get severe pathology. The same is true for sugar. And so many other metabolites can be used as markers of disease. And this is a really powerful area of research that is only beginning to be recognized, and CSU is one at the forefront of it. What is important also is that these metabolite molecules are species independent. So it's the same in the mosquito or the human or the mouse. So it really provides a powerful way for us to look at disease in different organisms. And the most important thing to understand here is that these metabolites are what your drugs and inhibitors are. So any drug is literally a metabolite, and it causes a change in your body to prevent disease. And so by looking at what your body naturally produces, we're actually identifying natural drugs that we can then use to alter the biochemical pathways in your body, and so alter how you respond to infection. So one of the areas that we are trying to pursue right now is a systems biology-based therapeutics. And that is looking at the organism as a whole and looking at, if you take two drugs, will there be an interaction between the two drugs? Do they get broken down in your body? And if they do get broken down in your body, how does that impact your physiology? What happens when you have co-infections? So a lot of problems with COVID-19 comes because you're getting pneumonia because of a bacterial infection or other co-infections together with the viral infection. And how does your body deal with that? We can study this, and we can preempt it and prevent it by understanding how this happens. We can also understand the microbiome, which is what you alter when you change your food habits or you take probiotics. And how does that alter infection or infectious disease? So what we think is the future, and we are very much, CSU is at the forefront of this future in infectious disease, is that every individual should have a personalized medicine repertoire. So maybe a drug does not work for one person or the second person, but the drug might work for the third person. And so we call it personalized medicine. This is where we think the future is going, and where, as I said, we're very much on top of it. We have recently formed the Center for Metabolism of Infectious Disease. I'm the co-director with my colleague, John Belisle. And we hope to study some of these interactions using these wholesome systems biology methods, where you look at the whole organism and look at infectious disease in a much more a broader view, so that we can prevent some of the complications we're seeing with COVID-19. So with that, I want to highlight some of the contributors. So the antiviral testing program was initiated by myself and Brian Geis, who's a colleague in the microbiology, immunology, and pathology department, but also with a tremendous amount of help from the Office of the Vice President for Research, where they donated David Patterson to us to help us with a lot of the legalities associated with antiviral testing. He's also a great advisor that helps us develop this program. I have 4 great students who have donated their time, working about 16 hours a day to really test these antivirals since probably March. Elena Liang, who was an undergraduate with me in my lab and now is a graduate student in the MIP department. Gabriela Ramirez was also an undergraduate with me in my lab and now is a graduate student in the Cell and Molecular Biology program. Carly McAllister was a master's student in my lab and now has been promoted to a research associate in my lab and is currently running the antiviral testing efforts. And then Laura St. Clair is a graduate student in my lab who's also helping Carly with all of the antiviral efforts. This effort would not be possible if it did not have funding. So I want to acknowledge the funding that has been provided by the Office of Vice President for Research at CSU and also the Department of Microbiology, Immunology, and Pathology. I also was the recipient of the Becher Foundation COVID-19 Research Innovation Grant that really helped to accelerate this antiviral testing facility and the contributions that we have done. So with that, I'm happy to take questions and I will stop sharing.
Cool. So the very first question, and feel free to write in more questions as they're coming in, was what has the process been like working with the virus hands-on?
Right. So this was, it was a really stressful time during March through, I'd say, May, because not only did we have to set up the facility, although we have state-of-the-art biosafety level 3 facilities, not every room was set up to study SARS coronavirus because you have to wear the suit. And so we needed to convert certain hallway spaces so that we could walk with the suit and some of the virus samples from one room to the other. The department and all of my colleagues were really supportive in that. And so we were able to set that up pretty quickly. And then we had to train ourselves to really understand this virus because my lab had never been working with this virus before, neither had any of my students. And so it was exciting, but it was also really tiring because you literally worked 16 hours a day trying to understand how to grow the virus, what cells to use, what conditions to use, and then how to test these antivirals. But it's been fruitful, and I think we're excited that we were able to contribute.
Cool. The next one is, and I'm not entirely sure if they heard the answer or not. What's the difference between a therapeutic and a drug? Or what's the basic difference between a therapeutic and a drug? Or the difference between the vaccine work at the SoloVax project and your therapeutic work?
Right. So vaccines are basically either components of the virus or whole virus that has been killed or attenuated. And so you get that to trigger your immune response so that the body remembers that it's seen something like the virus. And so when it really gets an infection, it's able to respond really fast and mitigate the infection. A drug or therapeutic is basically a chemical that you take after you're infected. And sometimes you could take it before as well. And mainly it's to inhibit the virus from its ability to replicate. And so what you're trying to do is within 24 hours of your symptoms, you're trying to reduce the amount of virus that's replicating so that your immune system can take over. So it's literally like trying to help your immune system get there faster by reducing the ability of the virus to take over your body. Does that help?
I hope so. And that's actually all of the questions that I have. So with that, I want to thank you so much for doing the presentation, and thank you everybody again for making it tonight. You get 20 minutes back of your day. So if you don't have any other questions, feel free, you can log off. We are recording this, so we will be putting this on our YouTube channel. Just go to YouTube and then type in CSU Alumni Association, and you can find all of our videos from the past as well as this video as well. But with that, thank you again, everybody, for making it, and go Rams.
Thank you, everyone, and thank you, Corey.
And thank you as well. That was fantastic.
Thanks.